A very good morning to everyone. I, Manali Jadhav, on behalf of SEP CRNTS IIT Bombay, take this opportunity to welcome you all to today's webinar on High Resolution Liquid Chromatography Orbitrap, titled Post-Translational Modification Proteomics Employing Dual and Hybrid Fragmentation Principles Coupled to Orbitrap MS. At the outset, I will provide overview of SEF, brief introduction of today's speaker Dr. Sarvanan Kumar, followed by his presentation on the topic, and at the last question and answer session. I request everyone to post their question with their name in question and answer box, which will be answered towards the end of the session. Sophisticated Analytical Instrument Facility, also known as SEF, was inaugurated by our late ex-Prime Minister Shri Morarji Desai in 1976. Project CRNTS was also started in 2006 with initial funding from DST. In 2008, SEF and CRNTS got merged under one umbrella headed by one in charge. Professor Anil Kotantharil is currently heading our SEF CRNTS department at IIT Bombay. 16 major equipments and 5 under CRNTS. CRNTS being academic wing also has PhD programs, 80 plus students guided by around 65 faculty members of other department. 2,402 users registered and 20,861 samples were analyzed during financial year 2018-19. The Department of Science and Technology, DST, has set up sophisticated analytical instruments facilities, SAF, in different parts of India. The main objective of setting up these facilities were to provide guidance in acquisition of data using sophisticated instruments. SEF at IIT Bombay has several analytical instruments and provides measurement services to IIT Bombay users and to users from other colleges, universities, national laboratories, and various industries as well. DST has provided funds for following instruments, out of which 3 liquid chromatography mass spectrometers have been highlighted. Out of these 3, 2 of them are high resolution mass spectrometers, QTOF and Orbitrap. These are instruments which come under the central facility funded by IIT Bombay. Dr. Mayuri Gandhi is the in charge of all mass spectrometers in-housed at SEF CRNTS. To know more details about SEF CRNTS, please visit our website sef.iitb.ac.in. It gives me great pleasure to inform that in spite of 17 SEFs funded by DST, we have users and researchers coming to our SEF from different parts of India and Nepal. SEF is not just the center for research, but we also encourage people to visit our center to help them understand and demonstrate how our facility works. We encourage visitors from industries and universities. We also have a LinkedIn page of SEF at IIT Bombay, and we would be happy to connect with you on LinkedIn. We have in all lined up 12 webinar series on different topics. You can find the details of upcoming webinars on our LinkedIn page also. I once again take this opportunity of thanking everyone for joining today's webinar. All our team members would be happy to welcome you to SEF CRNTS and help you with your research and development work. It gives me great pleasure to welcome Dr. Sarvanan Kumar. Dr. Sarvanan Kumar is currently associated with Thermo Fisher Scientific India Private Limited. He has gained his initial training at the Proteomics and Structural Biology Division, Institute of Genomics and Integrative Biology, New Delhi. He has done his PhD from International Center for Genetic Engineering and Biotechnology, ICGEB, New Delhi. He has more than 11 years of experience in the field of mass spectrometry, protein biochemistry, and analytical techniques. He has co-authored 20 plus international publications. He is currently leading the team specialized in biopharma, proteomics, and small molecule metabolomics. He is closely working with leading industries and academic institutes for developing LC-HRMS methods and workflow solutions for challenging problems. Now, I take this opportunity of requesting Dr. Sarvanan Kumar to present his slides to everyone. Over to you, sir.
Thank you, Manali. Thank you, Team SAFE IIT Bombay, So today's session, it will be on PTMs, the post-translational modification proteomics, employing dual and hybrid fragmentation principles. Like I'm sure like most of you must be aware, like the so-called mass spectrometry, it is all about gas phase behavior or the gas phase dissociation behavior. So the so-called gas phase behavior, we call it the intact MS or the MS1 approach. The so-called gas phase dissociation behavior, we call it the Msms approach, right? So that's all mass spec is about. So today's session will be on this particular so-called dual and hybrid fragmentation. So I'll be explaining about the importance of different fragmentation. I mean the gas phase dissociation principles. Specific emphasis will be on PTMs, post-translation modified peptides. So how these fragmentation principles are important and how are we trying to combine them? There are various means of combining fragmentation principles. So how are we combining them and how is that we are trying to improve the quality of information that we will be able to retrieve from a given sample. So the contents of my today's session are briefly listed down here. I'll start with the principle of Orbitrap. Then we'll go through the basics of protein modifications, and then the actual challenge, the ionization of modified proteins or the peptides. Then we'll go into the actual context, the peptide-centric approaches, wherein we will focus on the different fragmentation techniques, fragmentation principles. I'll also touch upon the different experimental design factors, real time, how should one design the experiments, and what to expect, what not to expect, a little bit on the computational aspects as well, and then we'll summarize the session. To begin with, currently we are in slide 4. This particular slide, it explains the working principle behind the Orbitrap mass analyzer. I'm sure some of you must have attended my earlier session. I'll just explain you the brief principle of behind this particular analyzer. The barrel-like structure that you see is the Orbitrap analyzer, and then there is a central spindle-like structure that is the central electrode. The rings that you see, three different color rings, those are the ion populations. So in principle, ion populations are moving around the central electrode, and they are moving with certain frequency. This frequency of movement, it generates image current. The image current is later being transformed using the algorithm FT, Fourier transform. Ultimately, the output that we see is the spectral plot, M by Z spectral plot. So this is how the Orbitrap mass analyzer detects our samples or the analyte ions of interest and generates the M by Z spectral plot. Now let's go into the next slide. Protein modifications. There are different kinds of modifications, but the one that is being most commonly explained or debated in scientific literatures are the PTMs, post-translation modification. Nevertheless, there are also other modifications that are of equal importance. Apart from PTMs, we do have co-translation modifications and of course in vitro modifications. So these are the three different modifications that one should anticipate when they are trying to analyze samples from biological source. Right. So this particular slide number 6, I'm just trying to show you the different, it's just a representative list. It is not an exhaustive list. So you can see there are different kinds of modification that is possible. Like in lysine, succinylation is possible, monomethylation, dimethylation, acetylation. Similarly in tyrosine, phosphorylation is possible, nitration is possible. So these are the different kinds of modifications, citylation, hydroxylation, and of course glycosylation. So these are the different kinds of modifications that one should anticipate in a given biological sample. Right. Now comes the challenge. So what is so special or what is the critical aspect when it comes to modified peptides or maybe modified proteins? It could be PTM, post-translation or co-translation, whatever it is. What is the challenge? Just to explain that particular challenge, I just want to show you this particular slide. So this slide explains the basics behind electrospray ionization mass spec. So I'm sure most of you must be familiar and habituated with ESI. In fact, Manali had showed you a table in which they had the different kinds of mass spectrometers. Among them, 2 ESI based instruments are also there. Even our Orbitrap instrument is ESI based. Now let's go into the actual challenge part, right? So in this particular slide, what I am trying to show you is it is a model, it's a theory. Theory behind how a electrospray ionization principle happens with regard to modified and unmodified peptides. That is what has been explained in this particular slide. If you see this right hand side panel, there is a big circle like thing, right? That circle represents the droplet. Whenever you infuse a sample or inject a sample through LC. So we use LC as a front end, right? Liquid chromatography. It could be a nano LC or a UHPLC. This is the droplet that forms at the emitter tip. This structure that you call, this is the emitter, ESI emitter. So in this droplet, what are the things that we can expect? Obviously samples, peptides. Apart from that, you have the solvent droplet, ACN. In addition to that, we have the ion pairing agent, like say formic acid or TFA or any other ion pairing agent. Now, what is important is that how are the analytes dispersed, distributed within this droplet, this circle like thing. One important aspect that we need to note down here is in any sample from a biological source, from a cellular source, the stoichiometry of modified versus unmodified peptides. I mean PTM peptides versus non-modified or unmodified peptides. So the PTM peptides will always be low. Point number 1. Point number 2, whenever a peptide backbone is modified by a modification, like say post-translation, like say glycosylation. This kind of a modification, it adds hydrophilicity to the peptide backbone, right? So having said that, now we have a mixture of peptides which are hydrophilic or at times zwitterionic. They don't carry a null charge. So we have this kind of a peptides. At the same time, we have peptide backbones with positive charge. At the same time, they're hydrophobic. So now all the hydrophobic peptides, the so-called unmodified or the non-modified tryptic peptides, they will all partition. They will localize themselves towards the end of the solvent droplet, the surface region. What about this modified peptides, like say glycosylated peptides? These are hydrophilic. Sometimes they don't carry a net charge, zwitterionic. So in such cases, those peptides, modified peptides, they will partition, localize themselves in the core region, center region. Now comes the challenge. When we apply a high voltage, all the peptide ions that are there in the surface, they readily get nebulized and they tend to get detected in the mass spec. Having said that, peptides, unmodified peptides will have very higher intensity. Now, what about these peptides that are localized in the center, modified peptides? Unfortunately, their intensity will be very less because the stoichiometry of those peptides are less. Point number 1. Point number 2, their amenability. They are not readily exposed to the nebulization process. Because of that, the unmodified peptides gets the priority and the modified peptides, they will get the next priority. Therefore, their overall intensity will seem to be less in a given spectral plot. So this is the actual challenge. Therefore, it is not only the complexity of the sample. You know, it has a peptide backbone, it has a glycan backbone, not just the complexity of the structure. It is also the complexity that one should anticipate during the ionization process. So these are something that we need to keep in mind when we are designing a PTM kind of a sample. So now let's go to the next slide. All right. So we know that stoichiometry is not in our favor. We also know that ionization is not in our favor. So how do we make it favorable? So that is what is being explained in this particular slide, slide 8. If you can see the point number 1, chemical tagging. This is nothing but derivatization, right? So it can increase the overall analyte charge density. or the gas phase basicity. Thereby, you can ionize the molecule much more efficiently as compared to the non-derivatized or non-tagged sample. That is one way of increasing the amenability of PTM modified peptides. Point number one. Point number two, use of nanospray. So whenever we deal with PTM samples, like say phospho, glyco, or even ubiquitination or myristoylation, these kind of samples, we always recommend using nanospray. So what is a difference? If at all you guys get any chance, maybe after this COVID lockdown, you can book a slot and you can have a look at the instrument, and Manali can even show you hazy spray, right? The UHPLC spray, droplet size will be bigger because of the flow rate, 0.2, 0.3 ml per ml per minute. Whereas nanospray, it will be the droplet size will be less, nanoliters, because it's all in nanoliters per minute. Therefore, the overall droplet size, if you would have seen in my earlier slide, that blue color circle, the overall droplet size will be minimum in nanospray. Having said that, you can have a control, reduce the effect of the ESI partitioning, the droplet partitioning effect. Thereby, you can also increase the possibility, amenability of the hydrophilic peptides, which is nothing but the PTM peptides. So these are some of the ways wherein you can minimize ion suppression of PTM modified peptides. These are the things with regard to mass spec. Now let us see a little bit of the wet lab. Before going into the mass spec, can we do something? Yes, of course. By enriching the modified peptides. It is not mandatory that you enrich samples to get it identified, but still we recommend it. We recommend to enrich phosphopeptides, glycosylated peptides, right? Purely to take care of the stoichiometry, so that we will be able to selectively enrich the modified peptides, and in other words, we will be able to deplete the non-modified peptides. That is the logic. And the other thing is fractionation. Of course, fractionation will help you to reduce the complexity. So these are some of the ways wherein one should, one can plan their experiment so that you get a very good PTM results, PTM modified peptide results. Right. Now let's go to the actual challenge with regard to sample. So far we had seen with regard to instrument, what could be the challenge one should anticipate when it comes to ionization. We also saw with something with regard to the wet lab, what kind of a stoichiometry one should anticipate, modified versus non-modified. Let us add little bit depth on the peptide structure. What kind of complexity we should anticipate when it comes to a spectral plot of a modified peptide. That is what I will be explaining you in the next 2 or 3 slides. Now, if you see in this current slide, slide 10, this top panel, left-hand side, this corresponds to the peptide Msms spectral nomenclature, the so-called A, B, C, X, Y, Z ion series. I'm sure you must have gone through Jonal Hunt nomenclature or Claus-Beiman nomenclature. That is what we use for peptide sequencing. So this is what it is. But in reality, this is what the right-hand side panel, what you see is a reality. It is never as simple as this. You don't simply get X1, X2, X3, Y1, Y2, Y3. Similarly, A1, A2, A3. It is never disciplined. If you can see this one left-hand side, it is very simple. Peptide fragmentation. Whereas the right-hand side that you see, this is the actual thing. Apart from A1, B1 or Y1, Y2, you do tend to see water loss, ammonia loss, all these kind of side chain losses, neutral losses are also expected. All these things together will add to the complexity of the spectra, the number of spectral lines that we get upon gas phase dissociation, right? So now let's go into this bottom panel. If you see, I have just plotted A, B, C, X, Y, Z, right? These are the 3 ion series. In fact, 2 pairs of 3 ion series from a given peptide sequence, peptide backbone, plus these ions, ammonium ions and internal ions. So these many ion populations are expected from a non-modified tryptic peptide or any other enzyme-lyased peptide. right? Point number 1. Now let's go to the next slide. Currently we are in slide 11. Earlier slide was from peptide Msms nomenclature, or the ion series we get from peptides. This particular slide, it is from the glycan. So this is a glycan structure. Again, these many ion series are expected if you are trying to analyze only a glycan, not a peptide, only just a glycan. Now let's go to the next slide, slide 12. So if you see bottom panel, the green ones, this is a peptide one, and this is a glycan structure. So in a given spectra, in a given sample, if you fragment a glycopeptide, you should anticipate these many ion series. So now you can visualize the number of spectral lines that one should anticipate in a given Msms spectral plot of a glycosylated peptide. So all these things are going to increase the complexity of the data. So now let's look at the choice of fragmentation. So since that we have a complex sample, complex analyte, we need to have, apart from the conventional fragmentation, like say HCD, CID, we should also have the additional fragmentation in place. So that is what is being explained in this particular slide. So this particular slide shows you this is a peptide backbone. Assume this would be an asparagine residue, and this is an end-linked glycan that is covalently modified onto the asparagine, nitrogen on the asparagine backbone, right? So if you see, it's these many fragments are expected, and the red one corresponds to HCD, collision induced dissociation, or the high energy collision dissociation. The blue one corresponds to the ETD, electron transfer dissociation. So the bottom line is that if you want to characterize a given glycopeptide backbone, you should employ both these kind of fragmentations, HCD or CID, at the same time ETD. So if you see in the bottom panel, I have written ETD-SA, which means ETD followed by supplemental activation. At the same time, I have written ET-HCD, which means electron transfer dissociation, high energy collisional dissociation. So this is the essence of today's session. I was talking to you about dual fragmentation. This is dual, and this is hybrid. So in the subsequent slides, I will explain you further what is this actual dual and hybrid means. All right. So slide 14. So here I'm just trying to show you the schematics of different commercially available Orbitrap platforms. The top panel, it's the Benchtop Instrument. This is what you have, top one left hand side panel. This is what you have in safe facility. The bottom panel is the Hybrid Instrument. The difference between top and bottom is, in top panel you can do only HCD, only one type of fragmentation, high energy collisional dissociation. In bottom panel you can do multiple fragmentations. That is what has been highlighted. In addition to HCD, you can also do CID. In addition to HCD and CID, you can also do ETD, ET-HCD, ET-CID, UVPD, so on and so forth. So different kinds of fragmentations are possible, right? All these things are just to increase the analytical capability, so that irrespective of the sample complexity, in today's session we are talking about PTMs. So there could be different kinds of complexity, right? So irrespective of the sample complexity, we will be able to get the best possible result. So that is the advantage of having the Hybrid over the Benchtop Instrument. As simple as that. All right. So where does these fragmentations actually take place? So-called CID, HCD, all these things, ETD, these things. So if you see in this particular slide, so HCD takes place here in ion routing multipole, right? ET-HCD, it also takes place in ion routing multipole. CID, ETD, PTCR, all these takes place in ion trap, right? And UVPD, it takes place in the rear end of the instrument. So these are the different fragmentation techniques, and we have dedicated components, hardware components, where these fragmentation techniques will be actually happening, right? And this is an earlier instrument. This is a hybrid instrument. Before Hybrid, we had the hybrid, and these are the different components, like say ion trap where CID takes place, and then octopole cell where HCD takes place, and then we have a dedicated ETD cell. But this particular instrument is no more. It is, it's absolute. It's just for your information. And this is the Benchtop Instrument. and it has only one fragmentation technique, that is a HCD cell, and that HCD takes place in this octopole cell. Right. Now let us look into a little bit on the what are the differences between CAD, HCD, ETD. Once we know the actual difference between the independent fragmentation techniques, I'll tell you about the dual fragmentation and hybrid fragmentation. It will be easy for you to understand. Now, CAD. CAD is nothing but the collisional induced dissociation. It takes place in the ion trap component of the mass spec instrument, Orbitrap mass spec instrument. Now, what is the actual thing? So, collision is induced through multiple low energy collisional challenge. So, there are, like say, you can induce collision through single channel or multiple channels. Point number one. Point number two, you can add more energy in terms of electron volt, or you can add less energy. So in this case, it is multiple low energy collisional channels. That is what is being employed onto a peptide backbone. And the source of collision is helium. So I'm sure you know what is collision, right? So we have a component, a hardware closed cell. That closed cell is filled with some kind of preferably a inert gas or a less reactive gas molecule. Into that you put the sample of interest. You allow them to collide. So that is measured in terms of electron volts. So in order for the collision to happen, in this case we use helium. In certain cases they use argon. In certain other cases they also use nitrogen. All right. So in CAD we use helium, and it is multiple low energy collisional challenge. So what happens is that the product ions, they fall immediately out of resonance excitation. So whenever we are subjecting a peptide backbone onto some kind of an energy, in this case collision energy, the peptide backbone will undergo vibrational excitation because it is absorbing energy. So when it absorbs energy from ground state, it will go to the excited state. So the consequence of that excitation is the vibrational excitation. That's what we call vibrational resonance. Because of that, you tend, whatever that happens, the consequence is what we got see is as fragment, right? So the advantage is that since we are using low energy collisional channels, there will not be any secondary fragmentation. Secondary fragmentation means, like say, you are subjecting a precursor to a fragmentation reaction. You expect only the genuine fragment out of the fragmentation reaction, the collisional reaction. However, since once the fragmentation has happened, the fragment will also carry some amount of energy, collision energy, right? Because of that, the fragment might tend to undergo one more level of fragmentation, which is not defined by you, which is not defined by the scientist. It is happening on its own, right? So that is called secondary fragmentation. That is not good, because software does not always know to calculate the secondary fragments. It will only know to calculate the primary fragments. Primary fragments are defined, disciplined, whereas the secondary fragmentation, it is more like a random, right? So the fragment still sustains the energy, and because of that, it is undergoing a secondary fragmentation reaction. So in CAD, that kind of a secondary fragmentation is very less, right? So the other thing is the modification. So there is a very good saying: the last bond to be formed is the first bond to get break. The last bond that is formed is the PTM bond, right? That's why it's called post-translational modification. After the protein backbone, peptide backbone is formed, after the translation reaction, the bond is formed, glycosylation bond or a phosphorylation bond. So that is the bond that is last formed. So that last formed bond is the first bond to get break. So that is what will happen when you are subjecting a glycosylated or a PTM modified peptide. The PTM gets dissociated first, followed by the actual peptide backbone. So that is one important thing that we need to keep in mind. Because of that, you will tend to see more PTM related information rather than peptide related information. So I have some spectral data wherein you will be able to understand what I'm actually trying to say, right? So this is the other thing is obviously we can direct it in ion trap or Orbitrap. And one more important thing, CAD, it has 1/3 cutoff. 1/3 cutoff means if you have a precursor of mass 1200, 1/3 of 1200, so that is 400. It will be a challenge for the scientist to visualize anything less than 350 or 400 m by z. That's called 1/3 cutoff rule. Now, unfortunate thing is that less than 400, less than 350, or less than 200, that is a mass range wherein we will see diagnostic fragments. For example, in case of tryptic peptides, 147, 175, lysine, arginine, right? In terms of glycosylated peptides, 163, 204. 163 it corresponds to exos, 204 it corresponds to XNAC. So all these diagnostic fragments, it will be there in the low M by Z. So therefore, monitoring the low M by Z is very, very important. In ion trap fragmentation, I mean CID, I'm sure you must have tried CID in other instruments also, like say MALDI, QTOF and other instruments. So it will be a challenge to visualize the low M by Z fragments. So low M by Z fragments are very important. So these are some of the pros and cons of ion trap. How ion trap collision induced dissociation works, what are the advantages and what are the disadvantages, right? Now let's go to the next slide. HCD. So HCD is high energy collision dissociation. It is an improvised version of CAD, right? Difference is that in earlier slide, these are multiple low energy collision challenge, right? Whereas here in HCD, it is high energy collisional channels, right? And the source of gas there it was helium, here it is nitrogen, right? So what happens is that higher energy is applied, however, the fragmentation happens through lowest possible energy channels. So this is a so-called gas phase dissociation pathway. So peptides they take a different pathway, fragmentation pathway. Glycans they take different fragmentation pathway. So are they navigating towards a high energy pathway or a low energy pathway? So that is what will determine the quality of the Msms fragmentation spectral plot. So in this case, it is high energy collision energy. However, the fragmentation takes place to lowest energy channels, and in some cases, high energy channels. So now comes the other thing. Product ions, the fragment ions, they will sustain the translational kinetic energy, and I mean the collision energy that is being absorbed by them. Because of that, you will tend to see secondary fragmentation, which we don't see in CAD in ion trap. Whereas here in HCD, yes, you do tend to see secondary fragmentation. And obviously bond. When it comes to PTM bond, they are labile, and in HCD it preferentially cleaves at the labile sites. So the PTM bonds will be dissociated first, followed by the peptide bond, right? So because of that, the more complete backbone sequence information, right? I mean the backbone sequence information in the sense peptide backbone information. So here we will be able to get good peptide backbone sequence information. However, since the PTM is lost from the site, it has been dissociated from the site, you will not be able to localize which site was actually modified. In peptides which has multiple PTM sites, there it will be very difficult for the scientist to annotate that this site was actually modified, because the PTM has already dissociated because of HCD energy, and so having said that, you will not be able to localize the modification site, right? The other important aspect is the low mass limit. There is no low mass cutoff here, which you saw in CAD. Therefore, you will be able to see all the diagnostic ions. So by now, I think you know how HCD happens, how CAD happens, what are the pros and cons of both CAD and HCD. Let's go to the next slide. So in this slide, you can see the head-to-head comparison of CAD top panel and HCD bottom panel. First thing, if you see this particular thing, low M by Z region, so you are not able to see any spectra here, right? This is CAD ion trap, and this is in HCD. You are able to see all the diagnostic ions or the low M by Z ions. Point number one. Point number two, you tend to see neutral loss. If you look into this slide critically, all are nothing but phosphorylated peptide ion series or neutral loss series. The neutral loss will have the intense peak. Because of that, you will not be able to get the complete peptide backbone information. That's what I showed you in the earlier slide. Now, if you see the bottom panel, you have the phosphorylated ion series. Apart from that, you do have other ion series. Therefore, your peptide sequence information will be much more complete, near complete in HCD as compared to CAD. So this is with regard to the CAD versus HCD, taking phosphorylated peptides as an example. Now let's go to the next slide. By now you know what CAD can do, what HCD can do, and what they both cannot do. They both cannot be used to annotate or to localize the site of modifications. So that is where ETD comes into principle, electron transfer dissociation. Both CAD and HCD, it is very hard. It's very physical process, collisional process. Therefore, you will not be able to sustain the labile modification on the site, on that particular glycosylation site or phosphorylation site. That's where you need a software fragmentation technique. That software fragmentation technique is ETD. So unlike CAD or HCD, ETD is more of chemical. It's like ion-ion reaction. Peptide cations plus fluoranthin anions. Right? These two ions react. The consequence of reaction is fragmentation, and the fragment is what we read as a spectral plot. Now, is it that easy? No, it is not that easy or that straightforward. If you see in this particular slide, so from left to right again, so this is the peptide ion, right? Peptide cations and fluoranthin anions. They are now made to react, ETD reaction. The consequence of reaction is we are able to see these many different populations. So whenever ETD happens, you tend to see ETD genuine fragments, Msms fragment, charge reduced precursors, and ET non-dissociated precursors. Apart from that, you also tend to see PTR, proton transfer reactants. So these are the different ion populations that one should anticipate when they are doing ETD kind of a reaction. Right? And from that, what is actually useful is the genuine ETD fragments. And that is what we use for annotating the site of a given PTM on a given peptide backbone. Right. So now this particular slide 22, it explains the ETD reaction. It's a single step fragmentation, and it is via gas phase ion-ion reaction. I mean peptide cations, and in our case fluoranthin anions. So it is relatively a democratic fragmentation, which means it is not driven by intramolecular energy levels, unlike CAD or HCD, which is actually driven by intramolecular. Peptide backbones, it absorbs energy. That excitation leads to fragmentation. It's more of intramolecular, within that molecule, right? So here it is not intramolecular. It's in fact interionic, in fact, cations and anions. And the direction is obviously either in ion trap or Orbitrap. The dissociation, it depends on the ion-ion chemistry, right? So what kind of charge is carried on by the peptide cations? Doubly charged are less amenable to ETD, whereas 3 charge, 4 charge, 5 charge are more amenable to ETD. Higher the charge, better will be a ETD reaction. The major advantage is that labile modifications, they are not cleaved, which means you will be able to localize and at the site of modification. That is a major advantage, right? And ETD dissociation, it depends on the charge density. That's what I told. Higher the charge state, better will be your ETD reaction. Now let us see some examples. Slide 23, it shows a phosphorylated peptide. It has almost 5 serines. Luckily, 3 serines are fragmented, whereas there are other 2 serines which are not fragmented, which is not part of the ion series fragmentation. Therefore, from this Msms spectra, you will know the sequence. However, you do not know which site is phosphorylated. Is it this or is it that? So now let's go to the next slide. This is ETD. Earlier say about CAD, this is ETD. So in ETD, we are able to see, yes, this is in fact this is a site that is phosphorylated, and this site is not phosphorylated. So this is the advantage of having ETD over CAD or HCD. Right. So that was phosphorylation, which was like say less complex modification, right? Hardly 1 phosphorus or 2 phosphorus molecule on a peptide backbone, right? Very rarely you tend to see 3 phosphorus molecule on a peptide backbone. So now let us see some complex PTM glycosylation. So top one is CAD, bottom one is ETD. If you look into the top one, very critically, we are able to get glycan composition. The preference is for glycan. glycans will be more as compared to peptide ions, right? So you will get glycan composition. However, you don't know the peptide sequence information, peptide structure. Now ETD. In ETD, you know the glycosylation site, which site is modified. However, you don't know the glycan structure or the peptide structure. There are hardly any fragments if you see, right? Now let's go to the next slide. HCD. HCD, you are getting very good fragmentation, right? You get both peptide and glycan. However, site is not known. So this is where we need to employ all the three different modifications. So for glycogen site, you need ETD. For glycan structure, you need CAD. And for confirming the peptide structure, you need HCD. So this is the kind of combinations that one should employ to get complete characterization of a given peptide backbone. In fact, a PTM peptide backbone. Now let's go to the next slide, slide 27. So here I'm just trying to show the influence of glycosylation on the Msms spectra. Bottom panel, there is no glycosylation. Top panel, there is only a minor glycosylation. I mean, there is only a glucanac and fucose. The blue circle denotes glucanac, and the green, the red triangle, it denotes fucose. If you see, this is the kind of fragmentation pattern that you see. No glycan and minimal glycan, and you are able to see the oxonium ions also. Now let's go to the next slide. It's the same peptide. Peptide sequence remains the same. However, if you see the glycan information, like the glycosylation is much more complex in the right hand side panel. As the glycan complexity increases, the fragmentation information that we get will be very, very less. Peptide fragmentation information. So that is why I put all the 4 Msms plot. No glycan, minimal glycan, and there is a abundant glycan. You just need to compare left hand side panel and right hand side panel head to head. So this shows the influence of glycosylation or glycan oxonium ions on an intact glycopeptide Msms spectra. Right? So now let's go to the next slide. This is just to sum up whatever that we were seeing so far. CAD, what CAD can do? CAD can give you glycan information, but no peptide information. ETD can give you glycosylation site. However, glycan composition, peptide composition, it cannot be attained. It might or might not be. HCD, you cannot get the glycosylation site. However, you will be able to get peptide sequence and glycan composition. So yellow is attained and red is not attained. As simple as that. So that is where we need to employ all the 3 different kind of fragmentations to get very good data. Right. Now let's see about the so-called dual or the hybrid fragmentation. Now, what do I mean by dual fragmentation? A precursor is being fragmented, fragmentation of a given precursor. It generates bunch of fragment. So the fragment will have some diagnostic features, diagnostic fragment. Presence of the diagnostic fragment will trigger one more fragmentation on the same precursor. That is called dual fragmentation. So fragmentation followed by a second fragmentation on the precursor level. This is called dual fragmentation or in tandem, one after the other. So for example, HCD triggered ETD. You do HCD on a precursor of 1200 M/Z followed by ETD on again 1200 M/Z. This is called dual fragmentation. Now what is hybrid fragmentation? We are fragmenting a precursor. It generates bunch of fragments. Now the generated fragments are subjected to one more level of fragmentation. The difference here is in the second one, we are fragmenting a fragment. Whereas in the first one, we are fragmenting only a precursor. Here we are fragmenting a given precursor using 2 different fragmentation principles. That is dual fragmentation. Whereas here we are fragmenting a given precursor using 2 different principles, but immediately one after the other. Here we are fragmenting a fragment, whereas here we are fragmenting only the precursor. That is the difference between dual fragmentation and hybrid fragmentation. I'm sure it will be a little bit confusing. I'll explain you with some of the slides here. Like say if you see this one, slide 31, this is an MS1 data. glycopeptide or glycoprotein data, right? So we are choosing this particular M by Z 645. Now it is fragmented. By fragmenting 645, we are getting these many oxonium ions. So you will, as a user, you will tell the software or the instrument, if you see these oxonium ions, you trigger one more level of fragmentation. That one more level of fragmentation is ETD. So ultimately, 645 is the M by Z, precursor M by Z. So the 645 is fragmented using HCD, at the same time using ETD. So this is a dual fragmentation, right? Now let us see how it actually happens. Top panel HCD followed by ETD. HCD triggered ETD, and bottom panel is CAD. So this particular event is triggering two other fragmentation events. So this is called the dual fragmentation technique. So from top panel, peptide information, second panel, glycosylation site information, third panel, we get glycan information. So this is dual fragmentation. Now let's see where we can employ. All these things can be employed in, say, not just for glycosylation, even for phosphorylation, sulfonation, nitrosylation, citrination, any different kinds of fragmentation technique. You can employ these kind of techniques to identify or characterize these kind of modifications. Now, these are the different modifications and its corresponding neutral loss or the diagnostic fragments, right? Just for your information. And now comes the importance of mass accuracy before going to the next one. So we have seen about dual fragmentation, right? Because in dual fragmentation, this mass is very, very important that you see here, because mere presence of this mass will trigger the next fragmentation technique, fragmentation on the same precursor. Therefore, we need to be very critical about the mass accuracy at MS2 level, right? So that is where Orbitrap is very important. Here, if you see, this is 84.08 and 84.04. They are near isobaric, right? But 84.04 is the actual oxonium ion, whereas 84.08 is a ammonium ion from an amino acid backbone, right? Therefore, it's very, very important to have mass accuracy at MS2 level. So all the high rated ones are the actual, the one that is sharing near isobaric with the black ones. Black ones are the oxonium ions, PTM diagnostic ions, and highlighted yellow ones are the near isobaric interferences. So this is where one needs to be very critical when they are designing an experiment, and choice of platform is very, very important. Right. So this particular slide, it shows the utility of oxonium ions and the importance of mass accuracy at MS2 level. It is essentially same. It is from a different paper. That is the only difference. Right. And this is the importance of resolution at MS2 level. If you see, if you can generate the data at a higher resolution, right, we will be able to separate 204.08 and 204.086. This is glucanac, whereas this one is a simple peptide backbone ion. So this is where resolution also is equally important. So both mass accuracy and resolution at Msms level is very important to unambiguously identify a given glycopeptide backbone or a PTM backbone. Right. Now comes the ET-HCD, the so-called hybrid fragmentation. Before going into this thing, what I'll do, I'll quickly show you another slide, so that will help you to understand the difference. I'll just unshare this particular slide, and I'll share another slide now. Right. So now I think you can see my slide. What I'll do, I'll put in the screen show mode, and yeah. So this particular slide 1, this shows the ETD, only one fragmentation, ETD reaction, right? So if you can see, this is MS1 scan, followed by isolation of the precursor of interest. So this red color dots that you see is the precursor of interest now. So they are now trapped in the ion trap. It's now going to the ion trap. Now what we'll do, we'll inject the ETD reagent, in this case fluoranthin anions. The blue color dots that you see, blue or the purple ones, those are the fluoranthan anions. Now the red ones and the blue ones will react. So this is the ETD reaction, peptide cations and fluoranthan anions. So they are reacting and the reaction is leading to the Msms one. So this is called ETD reaction. This is just one fragmentation. Now let us go to the next slide. ETHCD, hybrid fragmentation. What does it mean? Now let me do the same thing. We are getting the MS1 scan, right? Followed by isolation of precursors of interest. We are only isolating the same red color ones are the precursor of interest. So all these precursors will be trapped in the ion trap, followed by injection of the ETD reagent. Now the blue ones are again the fluoranthan anions. Just a minute. Now these two things, I mean the blue ones and red ones will be made to react, right? That reaction will lead to a fragment. That fragment is not been detected directly. It is now been sent here, ion routing multipole, and then it will undergo one more level of fragmentation. The fragment is fragmented again. That fragmentation is ETHCD. Ultimately, the ETHCD that we detect is this is the one that we are detecting. So that is the difference. So we are fragmenting here, that is ETD. The ETD fragments are passed on to ion routing multipole, wherein it is subjected to one more level of fragmentation. So that one more level is HCD. Therefore, we call it ETHCD. Ultimately, we are detecting the ETHCD fragment. So this is the difference between dual and hybrid fragmentation. In dual, it is precursors, whereas in hybrid, it is a fragment of a fragment. So what I'll do, I'll now go back to the earlier this thing. Yeah. So now we are in slide 40. So this is where we left. The benefit is that if you see the right hand side panel, HCD, ETD. The left hand side is ETHCD. When you are combining both these fragments, we are getting this kind of information. So that is the actual advantage of having ETHCD, the so-called hybrid fragmentation. Now, like say, these particular slides are all about how to optimize the ETD reaction, ETD reaction kinetics. We'll skip some of these slides because these slides will be too much technical. And only one thing I want to point out: why are we talking about ETHCD? Simply because of the reason, when I was explaining about ETD, I told you something called ETNOD, electron transfer non-dissociated fragments. What are they mean? What are those non-dissociated fragments? When we are fragmenting a given precursor, the precursor will lead to bunch of fragment. Now, each and every fragment should be independent spectral lines. However, in gas phase, these spectra, I mean the fragments, will tend to react with each other. Thereby, they form some kind of a non-covalent bonding. So there will be non-covalent bonding, non-covalent interaction between two fragments. Therefore, the two fragments will join hands and they will be seen as a very high M by Z. There, in order to separate them, we need to apply minimal energy. That minimal energy is called supplemental activation or the minimal HCD energy. So if you employ minimal energy, the fragment will dissociate again. The non-covalent interaction will be lost. Thereby, you will get independent fragments. So this will help you to annotate a very good Msms spectral plot. So that is the logic behind this so-called hybrid fragmentation, right? And that is what has been explained in this particular slide, right? So now this particular slide 44, it shows the influence of glycosylation on the ETD MS2 spectra. What the difference is that top panel is 3 charge, bottom panel is also 3 charge. Difference is that top is glycosylated, bottom is non-glycosylated. You can see the fragment rich information, what kind of fragmentation we are getting. So here charge is same, difference is glycosylation. Now let's go to the next slide. In this next slide, here if you see, it is +3 +4. It is the same peptide, same glycan structure. However, charge is different. So at a higher charge, you are getting better fragmentation. So this is what I told you about ETD. Higher the charge state, better will be your ETD reaction. So that is what has been shown in this particular slide. All right. So how can one improve the ETD reaction, ETD fragmentation reaction? One easier way of doing it is by using TMT. We in most cases we use TMT for quantitation, right? TMT can also be in use for ETD. So by adding a TMT label onto a peptide backbone, it increases the overall charge state density. Thereby, the 2 charge will be converted into 3 charge, or even at times 4 charges. By increasing your overall charge state, you are making the peptide more amenable to ETD reaction. So this is one way of doing it, one easier way of doing it. And so this is what has been shown in this particular slide. Top panel, there is no TMT. Bottom panel, we have added TMT label. So you can see the charge state, right? So all the 7, 6, and 5 charge state, the intensity has gone up. Whereas the lower charge state, the intensity has come down. Purely by the benefit of using TMT. Right. So in this particular slide, slide 48, what I'm trying to show is that top panel is CAD data. Bottom panel is ETD data. There is no TMT label on this peptide backbone. Now let's go to the next slide. Same peptide, same sample, we have added the TMT label. Merely by derivatizing the peptide backbone, you can see the comparison. Both CAD and ETD, the fragmentation has improved a lot. Thereby your annotation will be very, very authentic. You don't have to worry about your annotation ambiguity. 48, no TMT, and slide 49, there is TMT, right? So this is the benefit of derivatizing a peptide backbone, right? And what if you don't have ETD? So if there is no, I mean, of course, plan A is not working, then you obviously go for plan B. So that is what has been shown in this particular slide. So in cases wherein you don't have ETD, and if you have a complex PTM like glycosylation, what we can do is you enrich the glycopeptide of interest from the sample, and then the sample can be deglycosylated using PNGs. Now comes the criticality. So how do you do the deglycosylation? Usually we do all these enzyme reactions like say trypsin digestion, any phosphorylation, any kind of a enzyme reaction, we do it in normal water, O16 containing water. However, if you are planning to identify a glycosylation site, preferably do using a O18 labeled water. Why? Because during deglycosylation reaction by PNGSF, the asparagine amino acid gets deamidated. Deamidation of asparagine leads to 0.98 Daltons. This 0.98 Dalton mass increment can be used as a diagnostic feature to identify a glycosylation site. However, when we do digestion in neutral buffer, for example, ammonium bicarbonate, trypsin, all are like say 7, 7.2, 7.48 pH, right? So in this pH range, spontaneous deamidation of asparagine is known. Therefore, as a user, you will not be able to discriminate this asparagine is deamidated. Is it really glycosylated? Maybe or may not be. In order to overcome this ambiguity, we preferably do the deglycosylation using PNGs in O18 labeled water, because by using O18, instead of 0.98 Dalton mass increment, it will be 1.98 Dalton mass increment, because in place of O16 we have O18. So there will be a approximately 2 Dalton mass increment. So by doing so, we will be very sure, okay, this asparagine has 2 Dalton mass increment. That means it has undergone deamidation during the deglycosylation process, and it is not a spontaneous deamidation. By doing so, you will be able to unambiguously identify glycosylation site in the absence of ETD. So this is one way of identifying, characterizing a glycopeptide when you don't have ETD in your laboratory. As simple as that, right? All right. Apart from that, we can of course use this PNGs mediated O18 for quantitation also. Quantitation at glycopeptide and glycan level. So that is what has been shown in this data. Glycan quantitation and glycopeptide quantitation. Both these things can be simultaneously achieved. So this is just a representative workflow of identifying glycopeptide or a glycoproteome. In fact, this is a published paper. You can have a look at it in case if you are interested. So this is just for for the benefit of time, I'll just skip on these things. This particular slide is about etanercept. I'm sure some of you must have heard about this protein. It's a biotherapeutic fusion glycoprotein, very complex. You can see the complexity in this particular data. Left hand side, you see all the different possible glycosylation site and other modification sites. You expect both end-linked glycans, O-linked glycans. You also expect oxidation, deamidation. These many modifications are anticipated in a given protein backbone. And this is nothing but etanercept. So right hand side panel, we are employing these many strategies, different enzymes, different reagents to characterize this particular given protein backbone. And that is what has been shown in the subsequent slides, but we'll just skip it for the benefit of time. So I think by now you'll know what are the challenges one should anticipate when we are dealing with modified proteins or peptides, when it comes to stoichiometry, when it comes to ionization, and what kind of experimental design one should involve, like say enzymes or any charge addition agents, charge stripping agents, and what kind of fragmentation techniques, like say ETD, HCD, CAD, or ET-HCD, and what if ETD is not available, what is that we can do. So all these things have been discussed in this current session. So with this, I would like to conclude my session. If there are any queries, I'll be happy to answer.
Thank you, Dr. Sarvanan Kumar. Let us now begin with today's question and answer round. The first question is by Niranjana. She wants to ask why or where we use fragmentation of peptides. What are the applications of this method? Can we use this method for biomedical applications?
Yeah, of course. Biomedical in the sense, there are two things that we deal with biomedical. One is research, the other one is diagnostics. So in research aspects, yes, because most of the biotherapeutic proteins are glycosylated proteins. Most of the drug targets are again membrane proteins, which are again glycosylated. And some of the approved therapeutics are not just glycosylated, they are also fusion proteins. Therefore, we expect this kind of complex protein backbone, right? So one has to obviously employ these kind of combination of different fragmentation techniques to characterize these molecules. So this is point number one. Point number two, the other thing of biomedical is clinical diagnostic. In clinical diagnostics, you don't need multiple fragmentation. In most cases, it's just diagnosis. Just to diagnose based on a proven, approved biomarker. In such cases, targeted approaches, simply using CAD or HCD, will solve the purpose.
The next question is by Soumitra. He wants to ask, titanium dioxide is used to enrich phosphopeptides. What are the other PTM specific enrichment resins for glycans?
Yeah, so titanium, like say all the IMAC, right? Immobilized metal ion affinity chromatography. So titanium is one among them. Titanium, zirconium, and ion base, all these things can be essentially used for charge-induced peptide modifications. For example, phosphorylation, right? There are cases wherein people are used same titanium for glycosylation as well. Nevertheless, glycan-specific enrichment resins are also available. To begin with, lectins. Lectins can be used to enrich glycopeptides. Concanavaline A, WGA, VGM, agglutinin. These kind of lectin saccharose or lectin agarose resins can be used. Apart from this, boronic acid chromatography, it can be used. Hydrazide ions. So these are the different chromatography. Lectins, boronic acid, and hydrazide ion chromatography. These things can be used to selectively enrich glycopeptides. And in case of glycans, dissociate the glycans from the glycopeptide, pass them through a Dovex resin, positively charged Dovex resin. Thereby, you will be able to selectively enrich glycans. So these are the different approaches one can employ.
One more question by Anonymous. What is the best database for looking at PTMs?
As such, there is no one single database. Like say, if you have, anyway, you have PD, right? Proteome Discoverer. Most of the common known general modifications can be identified. phosphorylation, acetylation, methylation, myristoylation. These kind of known modifications can be identified. Like even the common known general glycan structures can also be identified. However, if you are dealing with unknown modifications, in such cases, we need to do a de novo sequencing kind of an approach. So apart from mass spec data, you should also have some wet lab data, like say an assay, biochemical assay, or a Western blot kind of an assay, which is specific for that particular modification, not for the proteins. Thereby, we will be able to identify and characterize that particular modifications. But a good starting point will be Proteome Discoverer. Apart from Proteome Discoverer, when it comes to glycans, you can use SimGlycan. That is another software that is available. But SimGlycan is a commercial one. It's like you need to pay a cost. And the free one, I can say Glyco Workbench. Glyco Workbench can be used. I mean, it's free of cost for academic users. Other than that, I can also suggest Protein Metrics, Bionic. So that is also very useful for this kind of a complex modification, like say glycosylation.
The last question, sir.
Yeah.
Can HCD or CID be used to find the site of PEGylation with PEG of large molecular weight by Hardik?
Site of PEGylation, right?
Yes.
Now, Hardik, let's say PEGylation is a like as the name suggests, right? Polyethylene glycol. Sorry. So it is polydispersed in nature, and this is nothing but polymeric units of ethylene glycol onto peptide or protein backbone. When it comes to identifying a PEG, it is not a straightforward approach. To begin with, you need to first generate an intact MS data, intact mass spectra. For that, we use triethylamine. It's an ion pairing agent. So when a polyethylene glycol is bound onto a protein or a peptide backbone, it tends to attract so much of protons. Therefore, you add charge stripping agents, deprotonation agents like triethylamine. Thereby, you will be able to capture the spectra, the data within M/Z range of a given mass spectrometer. So you will get an intact MS data. From that intact MS data, you will know the approximate molecular weight of the PEGylated peptide or protein. Point number one. Once you know this thing, we'll target that precursor. We'll fragment them. By fragmenting it, we'll know the actual sites which are non-exposed and the actual sites that are exposed. In most cases, polyethylene glycol, it is bound onto a peptide or protein backbone through primary amines, lysines, arginines, and N-terminus. So we will target those amino acid. Are these amino acid fragment ions visible in the fragment data? If yes, that means they are not modified. If no, that means there are chances of modification. Step number 2, you need to partially cleave the polyethylene glycol from the peptide backbone using chemical or hydrolysis means. We need to be very critical on this, because hydrolysis will also chop off the peptide backbone. So it has to be a mild hydrolysis, mild chemical reaction. Thereby, by doing this, we will be able to know, okay, this is a site which is actually modified by PEG. But my straightforward suggestion would be, in addition to using a polydispersed polyethylene glycol, consider using monodispersed polyethylene glycol. Monodispersed is homogeneous counterpart of polyethylene glycol. It will only give one peak, only one population. Whereas the actual PEG one, it is polydispersed, and it will give multiple peaks, multiple PEGylated population of the same peptide backbone. So these are things that you need to keep in mind.
So last question by Ms. Kriti Dalal. She wants to know, can you briefly explain about the chemical derivation of orthoglycans, how to analyze GAGs using MS? Okay,
O-glycans, right? So O-glycans is kind of like say it is less characterized, less experimented as compared to N-glycans. However, for O-glycans also, we can consider using the N-glycan derivatizing agents, for example, anthilinic acid, aminobenzoic acid, rapifluors. So these kind of N-linked glycan derivatizing agents can also be tested for O-glycans. Thereby, you will be able to increase the intensity of the O-glycans and also do some sort of a relative quantitation. Point number 1. Point number 2, how to analyze glycosaminoglycans. Now, glycosaminoglycan, just like we are employing different enzymes like for glycoproteins, glycopeptides, like trypsin, lysine C, PNGSF, PNGSA. Similarly, we also have enzyme mediated reactions and chemical mediated reactions for glycosaminoglycans. So depending on this thing, and ion pairing agents, we should also use different kinds of ion pairing agents. For example, there are, if I'm not wrong, TBE, tributylamine, these kind of ion pairing agents are available. So these things has to be used to characterize GAG molecules. Again, this has to be done step by step. So first intact mass data, spectral Msms spectral data, then derivatize based on this data. After that, we can do the subsequent characterization. That's how we do for glycosaminoglycans.
Yeah, that was the last question, sir. We are really grateful, sir, for answering all the questions. On behalf of SEF CRNTS, I would like to thank Dr. Sarvanan Kumar for interesting talk on post-translational modification proteomics employing dual and hybrid fragmentation principles coupled to Orbitrap MS. I would like to thank our head, Professor Anil Kotantharil, and our IT team members, Mrs. Nidhi Goel and Mr. Jagdeep Singh, for their continuous support in this webinar series. And last but not the least, I thank all the participants for attending this webinar and making this event successful. A link for the feedback form is shared in the question and answer section. Please do fill the form. Your valuable inputs are important to us. The upcoming talk is on structural characterization of soft matter using X-ray scattering by Professor Kamendra Prakash Sharma, and the registration link for this is now open on our website. I request you all to join us for our next talk. Once again, thank you all for making this webinar successful. Stay healthy and stay safe. Thank you. Thank you, sir.